Cellular metabolism begins with glycolysis, a process that breaks down glucose into smaller molecules. The final step of glycolysis is crucial, as it's where pyruvate, a key molecule for energy production, is formed. This transformation is catalyzed by the enzyme pyruvate kinase. What is pyruvate kinase? Pyruvate kinase plays a pivotal role in extracting energy from glucose, making it a vital component of the body's metabolic machinery. It is a crucial enzyme in the glycolytic pathway, where a series of enzymes break down glucose to produce energy. It catalyzes the conversion of phosphoenolpyruvate to pyruvate, generating adenotriphosphate or ATP, the energy currency of our cells. Without sufficient pyruvate kinase, energy production within the cell is severely impaired. This deficiency of the pyruvate kinase enzyme is called pyruvate kinase deficiency, or PKD. It is a genetic condition that can be inherited in an autosomal recessive pattern. That means that both parents must carry a copy of the mutated gene that have to both be inherited by the offspring. This can also occur as a specific gene mutation in the PKLR gene or the pyruvate kinase liver and red blood cell gene, which encodes the production of pyruvate kinase enzyme. Both conditions result in reduced production of the pyruvate kinase enzyme that contributes to PKD. What happens when there is lesser pyruvate kinase enzyme during glycolysis? During the last stages of the glycolytic process, pyruvate kinase is used to catalyze phosphoenolpyruvate into pyruvate and ATP. When there is less pyruvate kinase enzyme, the process of glycolysis is hampered, leading to inefficient glycolysis. This inefficiency disrupts the conversion of phosphoenolpyruvate to pyruvate that results into fewer net ATP produced after glycolysis. Since ADP is the energy currency of the cell, fewer net ADP produced during the cellular respiration process will result in some health consequences. On top of that, the inefficient glycolysis that occurs due to PKD also results in the accumulation of glycolytic intermediates, whose presence alters the cellular metabolism. This is called a metabolic bottleneck. This bottleneck exacerbates the problem of decreased ADP production, and the cell will struggle to meet its energy demands. In the case of red blood cells that require huge amounts of ADP to function properly, the lack of ADP causes prolonged energy deficiency and metabolic stress. A red blood cell that lacks ADP eventually succumbs to hemolysis or red blood cell destruction, as it can no longer maintain both its structure and function. In a person with pyruvate kinase deficiency, this often presents with the medical condition known as hemolytic anemia. This is a condition characterized by the premature destruction of red blood cells. A person who suffers from hemolytic anemia will struggle to deliver oxygen to their body due to the reduced red blood cell count. This sometimes results in the pale coloration of the skin due to low hemoglobin levels. Another possible consequence is jaundice, wherein the skin and eyes get a yellow coloration due to increased bilirubin from the breakdown of hemoglobin. The large amount of bilirubin extracted from the blood during the excretion process can also lead to darker colored urine. Another consequence of hemolytic anemia is the enlargement of the spleen. This occurs because the spleen has to work in overtime to remove damaged or abnormal red blood cells from circulation. The tissues of the spleen undergoes hyperplasia to enhance its filtering capacity. This enlargement of the spleen is called splenomegaly and contributes to anemia in a person with PKD. A person who suffers from PKD will need blood transfusion during severe bouts of anemia episodes and folic acid supplements to help with red blood cell production. In extreme cases, doctors may even recommend splenectomy to ease the reduction of red blood cells. Although PKD can lead to complications, Many individuals are able to manage their symptoms effectively with proper treatment and monitoring.